Right. So now in this lecture, we'll be discussing about the next portion, that is the next type of receptors, that is the receptor tyrosine kinases. So we'll start. Okay. So now it is the one second. Yeah. So now it is the receptor tyrosine kinase. Receptor tyrosine kinase. It's also known as RTK. So this receptor is also, it's a kind of a membrane receptor. So it's a plasma membrane receptor. It is a plasma membrane receptor. Now these kind of receptors, receptor tyrosine kinase, they have an intrinsic protein tyrosine kinase activity in their cytosolic domain. So they have, I will write in different color, they have a intrinsic protein, that is a tyrosine kinase activity, intrinsic protein tyrosine kinase activity in the cytosolic domain. Now the point over here is, so I'll just explain that. If you see the membrane, suppose this is the membrane, okay, and these kind of receptors, these are actually single transmembrane spanning receptors. So they have only these are not 7 pass receptors, they are only single pass receptors, right? So they will have only one membrane spanning domain, that is the alpha helix domain. So we know that. So we'll draw it in this way. Suppose this is the ligand binding domain, suppose this is the ligand binding domain, and this is the transmembrane domain. So I will just color it in different way. So this is the transmembrane domain, okay. This is the ligand binding domain, so this is the ligand binding domain, okay. And this one, this is the transmembrane domain, and the lower one, this one, it is the cytosolic domain, now the important point here is that this one is the membrane, suppose, okay, and this is the exoplasm, and this is actually the cytoplasm, right? Now there are important points we have to note, that this cytosolic domain, this has a particular tyrosine kinase domain, okay. So there is a particular tyrosine kinase domain, there is a tyrosine kinase domain present on the cytosolic part. So there is a tyrosine kinase domain where there can be, we'll see that in the next after some times, that there will be a cross autophosphorylation. So there will be a tyrosine kinase, so it actually phosphorylates the other different tyrosine residues, okay. So there is a tyrosine kinase domain, tyrosine kinase, it helps in the phosphorylations. This structure, we can say that there is a particular single, it is single membrane spanning alpha helix, there is a single membrane spanning alpha helix, right? Now another important thing is that the ligand binding domain, okay, the ligand binding domain over here, it's kind of, it is a cysteine rich, the ligand binding domain is actually cysteine rich. That's just an information. Now we need to know some important things, like normally when this receptor is not interacting with the ligands, they are present in their monomer, okay, they are present in their monomer. But when they actually bind with the ligand, they are present in their dimeric form. When they are actually binding with the ligands, so they are in their dimeric form and they actually come close to each other. Okay, so that is the case. So now first of all we can say that it's mainly the receptor of different growth regulators. Okay, so this receptor tyrosine kinase, examples of this receptor tyrosine kinase can be the growth receptors. Okay, growth receptors will be there. Okay, like EGF receptors, epidermal growth factor receptors. Okay, and insulin receptors will be also there. Okay, insulin receptors, just keep in mind that insulin receptors, they can be either of receptor tyrosine kinase type as well as they can be of GPCR type. So insulin has two types of insulin receptors are of two types. Okay, they can be either of RTK type or they can be either of GPCR type. So that's a very important information you need to know, that these insulin receptors, they can be either of, they can be of both the type of these receptors. Now the thing is, first of all here, so when there is ligand, so then what happens, this is the membrane and these receptors actually now they are normally present in their monomeric form there when there is no ligand, and when there is ligand they will come close to each other, and they will actually dimerize. So when there is ligand, then they will dimerize, now after dimerizing they actually do a particular different mechanism. I will do that after some points. I will give some important information, then I will explain the major pathway. Now what are the ligands, what type of ligands can bind to this kind of receptor tyrosine kinase type of receptors? What are the types of ligands which can bind? The first will be, which I told you, that can be EGF. They can be EGF. EGF is the epidermal growth factor. EGF is the epidermal growth factor. Second, they can be NGF. Okay, there can be NGF, that is the neural growth factor. There can be FGF, that is the fibroblast growth factor. There can be PDGF, platelet derivative growth factor. And there can also be insulin, which I told you previously. So these things are important, please remember these things. Any kind of growth factors will actually act as a ligand which can bind with the receptor tyrosine kinase. Now the thing is, the ligand binding site is actually the N terminal and the cytosolic site is in the cytosolic site is towards the C terminal. That is an important point, and we are also mentioned that towards the cytosolic site there is a particular tyrosine kinase domain. There is a tyrosine kinase domain. Okay, now let's move on to the mechanism. So when there is signal, so when signal is actually present, so when signal is present, so let's come to over here. Okay, so when signal is now present, then the receptors actually dimerize. The receptors dimerize. There is a particular, the activity of this tyrosine kinase domains towards the cytoplasmic site of the receptor, they become active. So the tyrosine kinase domain becomes now active. We will see a auto cross phosphorylation. We'll see an auto cross phosphorylation, okay, on the tyrosine kinase domain. On the cytoplasmic side, there is another kind of domain that is known as the phosphotyrosine domain. That is also present on the cytosolic side of the receptor, on the cytosolic domain of the receptor. They also get phosphorylated. I will draw that, don't worry. They also get phosphorylated. And when this both of these things actually takes place, then different downstream signaling molecules, different downstream signaling molecules can bind to the phosphotyrosine domain. Okay, phosphotyrosine domain by a particular mediator that is known as they have particular the phosphotyrosine domain binding area that is known as Grb2 by the help of Grb2. So I will show you that what is Grb2. Okay, so we'll see that what are the different kinds of downstream molecules. So one of the examples is Grb2, and they bind to the phosphotyrosine domain of the receptor by the help of SH2 domain. So I will show you what's happening over here. Wait. So the basic thing is that in case of this kind of RTK receptors, so I will draw that. So we'll draw the active condition. So it will be in a dimerized form. So this is the membrane. Suppose this is the membrane. Okay. Suppose this is the membrane. And suppose these are the receptors. Okay. So suppose if you draw the receptors like this, this is one receptor, and this is suppose another receptor. Okay, now it's in their dimerized form. Why? Because the ligand has come. So suppose this is the ligand. This is the ligand. It can be EGF, insulin, anything, and this is the receptor, and these are the dimerized form of the receptor. Now the thing is, here there is a tyrosine kinase domain. So I will write TTT over there. Okay, so there is a tyrosine kinase domain. So I'll just write over here. This is the tyrosine, this is the tyrosine, these are the tyrosine, and these are the tyrosine. And down the tyrosine kinase domain, there is another domain over here. This is the phosphotyrosine domain. So just write it down. This is the tyrosine kinase domain, and this is the phosphotyrosine domain. So these are the C terminal, these are the N terminals. Okay, so it's the cytoplasm over here. Now what happens when the ligand binds? Okay, when the ligand binds, the receptor dimerizes obviously, and there is an activity of the tyrosine kinase domain. What happens? It actually cross phosphorylates. So this tyrosine kinase domain it actually phosphorylates this, and this tyrosine is phosphorylated. Now in this tyrosine kinase actually phosphorylates this one. So it becomes phosphorylated. At the same time, this tyrosine kinase domain actually phosphorylates the opposite corner one. It also becomes phosphorylated, and this one also gets phosphorylated. So that is the, so this is actually the, this one is actually the, so there is no other enzymes coming. So this tyrosine kinase domain actually auto cross phosphorylating the other tyrosine chromates, and there is a cross phosphorylation, and it is done by the by its own tyrosine kinase domain. That's why it's known as auto cross phosphorylation. So this is the auto cross phosphorylation. And as a result of that, and after that, these phosphotyrosine domains are also get phosphorylated. Now this phosphotyrosine domain, when they get phosphorylated, now different proteins can bind over here. Now different proteins can actually come and bind over here. Okay, so there are different proteins, and one of the example is a Grb2 protein. Okay, they actually come and bind over here. Now this Grb2 protein, by the help of their particular specific they have a particular specific domain by which they actually try to bind. So they have particular domain which I have marked with this green color. So we can see this green color. So this protein is actually the, this protein is actually Grb2 protein, and this domain, by the help of which this protein actually binding with the phosphotyrosine domain of the receptor, this domain is known as the Sh2 domain. So Grb2 proteins, so different downstream signaling molecules. For example, Grb2 protein, we'll see that in case of the MAP kinase pathway, that this Grb2 protein is one of the important scaffold protein, right? So different downstream molecules, there can be Grb2 protein. We'll see in other examples of the downstream molecules can be Pi3 kinase, phospholipase C gamma. So we'll see in the other detailed pathways. So one of the example is Grb2, and by the help of its Sh2 domain, it binds with the phosphotyrosine domain, and as a result of that, the downstream signaling pathway is now carried out. Now the point is this receptor tyrosine kinase pathway. Okay, now the very important point, this receptor tyrosine kinase pathway, RTK pathway. Here, in case of the receptor kinases pathway, we'll see that the signal transduction is very fast over here. So the signal transduction is very fast, but there is no signal amplification. There is no signal amplification, and at the same time, there is no cross reaction. There is no cross reaction. In case of the G protein coupled receptors, we have seen that in case of the, when the G protein actually when these receptors actually activated, then actually it activates the adenylyl cyclase, and then the cyclic AMP synthesis becomes higher. It activates the protein kinases, and then it actually activates different transcription factors. Different genes are actually transcribed, different channels are being opened right now. So that's a kind of cross reaction, many functions, and the signal is actually amplified in different levels. But in this RTK pathway, you need to remember these 3 points. These are very important points because they will ask questions from this area. Signal transduction is very fast. We'll show you why the signal transduction is very fast as with respect to that of the GPCR. But there is no signal amplification. There will be a particular response. Suppose if there is insulin coming, so there will be a particular response. If there is EGF factor, so it will turn on, only the translations will be actually activated. So there will be a particular response, not a signal amplification, and there will not be any kind of cross reaction. There will be a unique response. If you are having particular, the amount of ligands you are actually interacting, that amount of response you will get. But in case of the GPCR, if you are actually if the interaction with respect with the receptor is very small amount of ligands are actually supposed to be small amount of ligands are actually interacting with the GPCR, then it will lead to signal amplification and the response will be large. But in case of the RTK, jitna ligand utna hi response, that's important, and the response will be very unique, so there won't be any kind of cross reactions. These 3 points are very important. They will ask questions from you. What is the difference between RTK pathway and GPCR pathway? They can give you different options, and these are the 3 options. And the reverse is case of the in case of the GPCR, right? Now, what are the different pathways? So opposite will be, इसका जो opposite है will be for G protein coupled receptors. Now, what are the different signal transduction pathway for where these RTK receptors are actually receptor tyrosine kinases are used? So we'll see that. So different signal transduction pathways. Okay. So now, first of all, in case of the, so there are, we'll study about 4 different pathways. One will be the RAS MAP kinase pathway. RAS MAP kinase pathway. Second will be the Pi3 kinase pathway. Pi3 kinase pathway, or it is also known as the AKT pathway. We'll also study about the Emptor pathway. And another will be the IP3 pathway. IP3 pathway. So now let's first start with the RAS MAP kinase pathway. So one will be the RAS MAP kinase pathway. Right. Now, in case of the RAS MAP kinase pathway, first of all, we need to understand many important things. Right. First of all, let's write in such a manner that if you just remember this chart, that will be enough. You don't need to go through different, you can actually check different images in the Bruce Alberts as well as in the Pathfinder Life Sciences, but I think if you just write this chart and just I think understand this chart, that will be enough for your competitive exams and also for interviews. The basic thing, I will just give you the main important point, that's enough. So suppose there is a EGF, this is the ligand, so it bonds with the RTK. Okay, so now the receptor will, so that there will be a receptor dimerization, so the receptor will dimerize. Very good. So now it will activate the tyrosine kinase domain. So activated tyrosine kinase domain, right? Now there will be auto cross phosphorylation. There will be auto cross phosphorylation of the tyrosine residues. Now the phosphotyrosine domain will also get phosphorylated. Now here through SH2 domain, the Grb2 protein will bind. The Grb2 protein will bind, right? Now please first of all write this chart, then I will explain you by drawing a diagram. That will be enough. First write this chart. Now to this Grb2 protein, another protein will bind, that is known as the Sos protein. This Grb2 protein is a kind of a scaffold protein. It's a scaffold, it's actually a mediator. This Sos protein is a kind of guanine exchange nucleotide factor, GEF. So it will actually somehow it will give the GTP to the particular protein. So there is a particular membrane spanning protein that is known as the RAS. RAS protein is present on the membrane, but it's present in its GDP bound configuration. SOS protein being a GEF, it will actually convert this RAS GDP to RAS GTP. RAS is also a G protein. When RAS GTP is being formed, then this actually activate another kind of a factor or a protein that is known as the RAF. RAF का full form है RAS associated factor. RAS associated factor, and this is nothing but a serine threonine kinase. It is nothing but a serine threonine kinase. It is nothing but a serine threonine kinase. Now this RAF, it now actually phosphorylate. They now actually phosphorylate another important protein that is known as the MEK. MEK. Now this MEK, now it actually phosphorylated another important protein. Phosphorylate another important protein that is known as the ERK. This ERK now phosphorylate. So this ARK actually enter into the nucleus and it actually also phosphorylate ELK1. Now this ELK1 in the nucleus actually activates different transcription factors and helps in the protein translation. So it actually helps in the gene expressions. What kind of genes? You have studied in the cell biology, in the cell cycle, like cyclin D, E and all. So it actually, so EGF actually acts as a mitogen. Now let's see the diagram. So we have actually made a particular flow chart. So you need to remember this flow chart. Okay, you need to remember this flow chart for RAS MAP kinase pathway. Now you will say, where is MAP kinase? So I will show you. This ERK is actually, it's also known as MAP kinase. Mitotic associated protein kinase, I think something like this. Mitogen associated protein kinase, like kind of this kind of thing. That's why it's called MAP kinase. MEK is phosphorylating MAP kinase. So it's actually also known as MAP kinase. So it's actually the enzyme. It's actually acting as an enzyme which is phosphorylating MAP kinase. So it's a MAP kinase. So RAF will be MAP. Okay. It will be, sorry, MAP, capital AP hoga. So MAP. Now this will be MAP kinase. MAP kinase, MAP kinase, and this is MAP kinase. This one is very important. These things are actually very important. So now I will show you a diagram, then you will understand. That will be enough. So let's do it in this manner. Suppose this is the membrane. Okay, and suppose this is the nucleus. Right. Suppose this is the nucleus. Now, and these are the receptors. So I will draw the receptor first. So suppose this is the receptor. Sorry. Okay. So this is the receptor. Now the ligand has been bound. So this is, suppose this is the ligand. So there is a ligand bound state. So in the phosphotyrosine domain, so in the tyrosine kinase domain, there is an auto cross phosphorylation, and these tyrosines are now phosphorylated. And the PTB domain or the phosphotyrosine domain, these are also getting phosphorylated. Now the Grb2 protein will come. The Grb2 protein will come, and it will bind by the help of its SH2 domain. So this is the Grb2 protein. This is the SH2 domain, right? Now what will happen? Now another protein will bind to the Grb2 protein, and that protein is known as the Sos protein. So this is known as the Sos protein. Now this Sos protein is acting as a guanine exchange nucleotide factor. This Sos protein is acting as a guanine exchange nucleotide factor. Now this guanine exchange nucleotide factor, I told you that is actually, now there is a particular membrane bound protein that is known as the RAS. It's a big protein. So I'll draw this. So suppose so this is the protein which is present over here. This is a big protein. So this is the RAS protein. Now it's present in its GTP bound form. Okay. So when it acts, when it interacts, so this GDP actually goes out and GTP comes in, and it is now bound in its GTP bound configuration, right? And this GDP actually goes out. Now that RAS GDP, to this it actually activates another important protein, okay, that is known as the RAF. RAF is also known as MAP kinase. It actually now phosphorylate another important protein. This is known as the MEK. So it gets phosphorylated. This MEK now phosphorylate another important protein. This is known as ERK. They get phosphorylated. MEK is MAP kinase, and ERK is MAP kinase. Now this ERK now actually goes into the nucleus. Now it is coming and going towards the nucleus. Okay. Now in the nucleus, suppose this is the DNA. Suppose this is the DNA. Okay. This ERK1, so the DNA is not looking good, so I'll just change it. So suppose this is the DNA. And now this ERK1, it now actually phosphorylate another important protein. It actually now phosphorylates another important protein, and that is the ELK1. And it now actually recruit different transcription factors, okay, different transcription factors, and they actually helps in the transcription of different genes. And in this way, this RAS MAP kinase pathway is being done. That is all about the RAS MAP kinase pathway. Now the point is this RAF protein, this MEK protein, and this ERK protein. Okay, so I will just draw it over here. This ERK protein, they also gets phosphorylated. Okay, now this protein, then it goes to the nucleus. These RAF protein, ERK protein, MEK protein, as well as this I can say that this MEK and ERK protein, they are actually bound with a particular scaffold protein which is actually holding these three proteins as well. This diagram is not good, so I'll draw it over here. So please see this. So I'm drawing this over here. The one that is another important protein. Okay, so it's like this. Okay, this is a kind of a protein where the RAF is bound, this MEK is bound, and this ERK is also bound. After that, this ERK1 gets dissociated and goes into the nucleus. There is a particular scaffold protein, okay, which binds these two, RAF, MEK, and ERK. This name is, the name of this scaffold protein is known as KSR. You don't need to know about the details of this, but the point is, so it's looking like this actually. So this is binding like this, this. The scaffold protein is actually holding these things. Now, as these proteins are being held by a scaffold protein, these proteins are near to each other. So these proteins don't have to find the other proteins to phosphorylate others. These proteins, they don't need to find the other proteins, because as they are attached in the same protein, so they actually phosphorylate or they can find the target very easily, because these are associated with the same protein. So that's why the signal transduction, because they phosphorylate this, so that's why as a result of that, the signal transduction is very much fast. That's why the signal transduction is very, very, very fast. And that's why we have told you that in the receptor tyrosine kinase, the signal transduction becomes very, very fast. Due to this scaffold protein, it's actually holding three different proteins. So in GPCR, they have to find the protein kinases. They have to find the protein kinase, then actually they get activated, and then this protein kinase goes to the Grb binding protein, then phosphorylates it, and then this Grb binding protein goes and recruits different transcription factors. So there is a long time, but in this case, as these proteins, main important proteins are actually bound with the scaffold protein, that's why the signal transduction is very fast. Okay, so that was all about the RAS MAP kinase pathway. Okay, now next is the second one, is the Pi3 kinase pathway. Pi3 kinase or AKT pathway. Now I will just write important points. You don't need to know, you don't have to actually know very details. They may ask question, they might not, but I'll just give you this point. This is an important pathway, but just remember that if questions are being asked from cell signaling, they will give you the important necessary informations. Okay, that is important, because in many colleges they actually don't teach the cell signaling. So in the question paper, if they ask question, so they will give you all the necessary information, and you have to just apply your logic. But we can't take any risk, that's why we are actually giving you all the important concepts. So now what happens, the same thing in case of the Pi3 kinase pathway up to the activity of the receptor. So I will not again start from the receptor binding. So the receptor ligand has bound and the receptors have dimerized, and then actually the autophosphorylation has been taking place, and then the phosphotyrosine domains are also being phosphorylated. So now we are having a configuration where having this situation where the receptors, where in the receptor the PTB phosphotyrosine domain is now phosphorylated, and to this here now to this phosphotyrosine domain this Pi3 kinase protein binds. Pi3 kinase protein binds. So in the previous case Grb2 protein was binding to the phosphotyrosine domain. Here the Pi3 kinase now here binds. Now when it binds, they actually phosphorylate PIP2 to PIP3. It was phosphatidyl inositol diphosphate to phosphatidyl inositol triphosphate. It acts as a secondary messenger. It also acts as a secondary messenger. Now what happens? So the basic thing is, now I have to draw few important things. So this is the case. Now this PIP3 has been formed, and they actually is bound to the membrane. So these are the PIP3. So I have not drawn the previous one. So this part as per the previous concept. So I didn't draw the previous one, the receptors and all. Just I am showing you the PIP3 portion situation only. So these are all the PIP3. Now different proteins bind to PIP3. So 3 different proteins will bind. So one is, this is one protein. Okay. This is another protein, and this will be another protein. So these are the Pi3 kinases, sorry PIP3. Now these are the 3 different proteins which are now binding. First, this one leftmost side, that is the Emptor C2 protein. C2 protein. Emptor C2. Middle one will be AKT. AKT, or it is also known as protein kinase B. Protein kinase B. Another protein which is actually binding to the PIP3, that is the PDK1. PDK1. So from here we are going to this area. Now what is actually happening? Now we can see that these PIP3 protein, now these different types of proteins are now binding to this PIP3. Very good. Now what happens? This Emptor C2 as well as this PDK1, they actually phosphorylate this AKT. This Emptor C2 and this PDK1 phosphorylates this AKT, and then this phosphorylated AKT, they become this, now this AKT which is phosphorylated, now it becomes active, and these phosphorylated AKT does many important things. You have to know few things, that it inhibits the apoptosis. So there is an important protein for apoptosis, that is called the BAT protein. It's a pro-apoptotic protein. It's a pro-apoptotic protein. Apoptotic protein. It inhibits the action protein. So as a result of that, there is no apoptosis. It actually favors the translations also. It also favors the translation. Okay. So these things are very important. So there will be no apoptosis. Sorry. So there will be no apoptosis, and there will be translation. There will be translations. So this was all about your AKT pathway. You need not know You don't need to know more details for your exams. You can know obviously for your own knowledge, but not for exams, because you have to know many things. Now another pathway. This pathway is the Emptor pathway. Now Emptor is known as the TOR. T is known as the full form is the target of rapamycin. Okay, target of rapamycin, because this rapamycin is one of an anti-cancer drug. It's an anti-cancer drug, and this anti-cancer drug actually targets this protein. That's why it's known as Emptor, target of rapamycin. It's an anti-cancer drug. Now suppose if in cells, suppose if the ATP amount is very high and the oxygen level is also very high, then like this same manner, this AKT kinase will be activated. Like the same manner is, suppose there will be a different growth factor receptors, then growth factor, then it will bind with the receptors, article receptor, then there will the autocross phosphorylations, the PTB domains will be also be phosphorylated, then this Pi3 kinase will bind to the PTB domain, and then it will convert the PIP2 to PIP3. The same thing will happen, and this by the same manner, this Emptor C2 will bind to the PIP3, APDK1 will bind to the PIP3, and AKT will bind, and they will phosphorylate this AKT, and AKT kinase will be active. Okay, very good. This is AKT kinase. Okay, AKT kinase. AKT kinase will be active. Now the same thing. So I'll not draw this previous portion. So you can draw that again, or you can't draw. You don't have to draw. Just understand the concept. So by the same manner, it will lead to the activation of this AKT kinase, and this is now active. Now when the AKT kinase is active, now it will phosphorylate an important protein that is known as the TSC1 and TSC2. You don't need to know the full form. Now normally what happens is that this TSC1 and TSC2, this TSC1 and TSC2, they actually inhibit an important protein that is known as the Grb protein. But when they get phosphorylated, okay, normally they used to inhibit the Grb protein. Okay, normally these used to inhibit another an important protein, that is the Grb. But when they get phosphorylated, they cannot inhibit. So they cannot inhibit. As a result of that, we can also write that this becomes activated, this becomes activated. So normally they inhibit, but when they are phosphorylated, they cannot inhibit. So Grb proteins are activated. Okay. Now when Grb protein is activated, it's a very important protein. When Grb protein is activated, it's a very important protein. Then it activates another important protein known as the Emptor C1. Here this was Emptor C2, this is Emptor C1. Emptor C2 activates the AKT kinase. Emptor C1 is one of the important protein downstream of the AKT kinase. So Emptor C1 now gets activated. This Emptor C1, it phosphorylates EIF4 binding protein. Now the important thing is that in the translation, in the eukaryotic translation, I think you have studied this eukaryotic translation, and you have known that there are different initiation factors in the eukaryotic translation, like EIF1, EIF1 alpha, right? There are different, you have to know this, it's very important. EIF1 alpha, right? EIF2, EIF5, 5 beta, 5b, okay? And there are different cap binding proteins like EIF4E, which is actually binding to the 5' cap. That is an important protein known as the EIF4 This is EIF4E BP. Then EIF4G, there EIF4A, EIF4B, EIF4E, G and A. You can call EIF4F. So these things are actually important. You have studied in the eukaryotic translations. I think so she has done this. So yeah. Now the point is there is an important protein. So this EIF4 is one of the important protein. We have studied in the translation that this is a kind binding protein. It binds to the 5' cap. There is an important protein, that is the EIF4E binding protein, and it actually binds to the EIF4E translation factor or the initiation factor. And when EIF4E is bound with this, when EIF4E is when it is bound with this EIF4E binding protein BP, it cannot perform its function because EIF4 is actually inhibiting the action of the EIF4E binding protein. EIF4E binding protein is actually inhibiting the action of the EIF4E normally when there was no phosphorylation. But when it gets phosphorylated, it cannot inhibit. As a result of that, it is activated EIF4E, and then translation takes place. So when this EIF4E, so if you actually, when this Emptor C1 comes and it actually phosphorylates this EIF4E binding protein, when it gets phosphorylated, then it cannot bind. So as a result of that, this EIF4E becomes free and it comes out and it can perform its function, leading to the translation. So phosphorylated EIF4E binding protein cannot bind to the EIF4E, and as a result of that there will be translation, because EIF4E is a cap binding protein. It helps in the eukaryotic translations. This Emptor C1, it will inhibit an important protein that is known as the ATG1. It will actually is an important protein. It will inhibit ATG1 protein, and there will be no autophagy. Just you have to know that Emptor C1 actually inhibits autophagy. So if there is high ATP and high oxygen condition, so there will be no autophagy because Emptor C1 will be prevalent. Okay, this will also activate an important protein known as the X6 kinase, and it will helps in the ribosome synthesis. And as a result of that, translation will be more. But if you have low ATP condition, that means high AMP condition and low oxygen condition, that is mean hypoxic condition, then AKT kinase is not active, Instead of that, another important kinase will be active, that will be the AMP kinase. This will be active. But this AMP kinase will not phosphorylate this TSC1 and TSC2. No, rather it will activate TSC1 and TSC2. So they will now inhibit the Rep protein. And if Rep protein is inhibited, so Emptor C1 will not be formed. And if Emptor C1 is not formed, so there is no one to inhibit the ATG1. There is no one to inhibit this ATG1. So as a result of that, ATG1 will be active and it will lead to autophagy. And also, as there is no Emptor C1, so there will be no translation. And there will be autophagy. You just have to know one important thing is that in the cell biology I have told you that LAMP, LAMP is important marker for the autophagosome, and you have to know that in hypoxic condition, that is the low ATPI or the low oxygen or high AMP condition, in this condition autophagy takes place. In these conditions autophagy takes place, and ATG1 is an important factor which helps in the process of autophagy. You don't need to know the details of autophagy. That is the case. Okay, and the last one is the this rapamycin drug, anti-cancer drug, it actually targets this mTORC1. Okay, now the last type of signal transduction, that is the fourth one. This is the Pi3 pathway. So it's very easy. It's same like that of the GPCR one, but just it's mediated by this Pi3 pathway. So the name of the enzyme is just different, baki sab same. So I will not tell you the first things that ligands will bind, then the receptor will dimerize, then there will be autocross phosphorylation. All these things will happen, and then the phosphotyrosine domain will be. So suppose this is the, so I'll just draw the final. This is the dimerized one. This is the membrane. Okay, and this is the, these are the also autocross phosphorylation condition, and these are the phosphotyrosine domain. These are also phosphorylated, and now another protein will bind. That protein will be phospholipase C gamma. In case of the GPCR, it was phospholipase C beta. Here it will be phospholipase C gamma, and they will also bind. They will also bind to the PTP domains by the help of the SH2 domain. Phosphotyrosine domain से वो SH2 domain से ये phospholipase C bind करेगा, right? And this phospholipase C will have the same function like P. It will convert the PIP3 to IP3, and DAG, it will associate with the membrane. It will actually activate the protein kinase C. Okay, and it will help in the metabolic regulations and metabolic and gene regulations. And you know that is IP3 will go to the ACR. Okay, it will release the calcium 2+ in the cytoplasm, and the same thing will take place, that reticulin, calmaluline proteins will bind with the calcium, and the same response. Okay, different roles of calcium will be there in the, different roles will be there in the cytoplasm for calcium. So that is it for the receptor tyrosine kinase pathway, right? Other important thing, just for note, you just write it. You will not ask questions from here. That in case of the, there is another kind of pathway called tyrosine kinase associated pathway. So here example will be JAK-STAT pathway. So important proteins over here, that is known as the Janus kinase and STAT. You can see Pathfinder Life Sciences the JAK-STAT pathway, but they will not ask questions. It's very easy. Instead of the autocross phosphorylation, this Janus kinase will bind to the tyrosine kinase domain. They will actually now cross phosphorylate, and the STAT proteins will bind. So I'll just explain it. So it's easy. It's very easy. So JAK-STAT pathway. Okay, so same thing, like suppose this is the membrane. Okay, and suppose this is the, these are the, like this, but they don't have the tyrosine kinase domain. Okay, so there is no autocross phosphorylation. Now what happens is that when the ligand binds, suppose this is the ligand is now binding, now there is important protein will come over here and bind over here, and now what will happen, these proteins will bind over here. These proteins are known as the Janus kinase. It's also known as JAK, Janus kinase, JAK. They will now cross phosphorylate. And the PTB Phosphotyrosine domain also becomes phosphorylated. And now what happens? Two proteins will come. These are the stat proteins. These are the stat proteins will come and bind to the phosphotyrosine domain, and these stat proteins become dimerized. The stat protein becomes dimerized, and this dimerized stat protein will enter into the nucleus, and it will help in the gene regulation. That is known as the JAK-STAT pathway. It's not the tyrosine kinase pathway, because it is tyrosine kinase associated, because there is no intrinsic autocross phosphorylation of the tyrosine. So there is tyrosine is present, but the tyrosine kinase domain is not present in the receptors. So these are tyrosine kinase associated receptors. So these receptors are not RTK. These are receptor. They are tyrosine kinase associated receptors. Okay, so that will be all. NF kappa beta pathway is important for interviews. They will not ask question from the in any exam. They will not ask question from NF kappa beta pathway. It's not actually important. Beta catenin pathway is also not important. You can see that for your own learning, but I'll say you that for exams, before the exam, you don't need not to check those things. In before interview, we can discuss important receptors of the important ligands for the NF kappa beta pathway, because this is very much related to the immunology sections. Okay, and because different responses are mediated by this NF kappa beta pathway, different natural killer neutrophil responses are being mediated by this NF kappa beta pathway. So I think for this competitive exams, this NF kappa beta pathway is not so important, but it is important, really important for your interview. So we will discuss those portions in that time. Okay, so up to this, that will be all for your cell signaling. Okay, please go through this Emptor pathway, MAP kinase pathway. These are super important. G protein coupled receptor. These are super important. Okay, please try to solve the different papers. And in TIFR, they will ask actually questions from here. Few questions are there in the previous papers, so you can check those and you can solve the questions from your MCQ books, and that will be all for this cell signaling. So by this we are done with this cell signaling chapter. I think that will be all. Thank you guys. Bye bye.